Namaste and welcome you all in our November month 2024 KSAS talk series. And we have again Dr. John Bogdan here with us the second time in this KSAS talk. Before starting this KSAS series, for those who are new here on KSAS platform, I want to introduce the Purvom School of Advanced Sciences, which is the Indian subsidiary of USA-based Institute of Advanced Sciences, Bachulanam Research Center and School of Indic Studies here in Mohanlalganj. KSAS is newly established research center with a vision to create a vibrant research, scholarship, and educational institution. We have few objectives with you can get the initial information about why have started this KSAS program and other KSAS series in Philosophy Cafe and other series. To develop the advanced research center involved in developing cutting edge biotechnology basics and applied research, promote holistic technology and the education. We have already introduced two national and international symposium here in KSAS Lucknow, develop fundamental theoretical and practical knowledge, both in science and humanities, investigate and promote traditional technologies scientifically, and it make available for the common people, especially for the rural areas, which the basic fundamental theme of Dr. Balram Singh, who is here, the president of INETS, to promote, to enhance the scientific temperament in village people of India, and also to engage in academic and scholarly communities. That's why we are organizing this. We are running here few projects in KSAS. I want to discuss few projects: therapeutic antibodies, treatment for spinal and cord injuries, project on finding better treatment for cancer using holistic approach, virus and bacterial engaging, development of charismatic delivery system, development of micromolecule based treatment options, developing our own protein expression system. In humanities field, we are running two projects: philosophy of existence, concept of Ramat, which is the social based order, economy of philosophy, natural world order, and the flickers of the century philosophers of India, which we have conducted a seminar on this topic 2 days before. We have here today with us Mr. John Bogdan. We have already uh, listened him in previous year on monoclonal antibody product development, past, present, and future. And this is the list, the speakers to whom we have invited previously in KSAS series. We have already invited 17 scholars from different fields at science, humanity, communication, and other fields, and took their thoughts. Today we have Mr. John Bogdan, who received a BS in biology from Rensselaer Polytechnic Institute and then PhD in microbiology and immunology from Sunny and Buffalo Jobs School of Medicine and Biomedical Sciences. From there, he went to on a perform a postdoctoral fellowship on DuPont Merck Pharmaceutical Company. After his postdoc, he became a research scientist in Kuwait, laterally vaccine and pediatric, and then the senior research scientist in North America, vaccine industry and Baxter Biosciences. He spent over a dozen years working on National Institute of Allergy and Infection Disease, NIAID, as a scientific review officer and program officer before starting his own consulting company. So we are here today with Mr. John Bogdan, and he will speak on the pathopsychological changes and immunological response to infections caused by Neisseria meningitidis. So, we, on behalf of INETS and KSAS, welcome you, Dr. Bogdan. Over to you.
Thank you very much. I'm going to bring my presentation up on the screen in a short minute. I just want to tell you a little bit more about myself. I'm located in Westminster, Maryland. I've lived in Maryland for about 25 years. Prior to um, restarting my consulting business, I was a senior advisor for the Department of Defense at Fort Detrick. And Fort Detrick works on all the category A through C pathogens that could infect the warfighter, like Ebola and monkeypox and a number of other different types of bacterial infections like plague, tularemia. So this presentation takes me back to my days in Baxter, where we actually worked on the meningococci. We were developing a Group C vaccine, and we were also interested in studying the porin protein as a potential carrier for glycoconjugate vaccines to replace tetanus toxoid or diphtheria toxoid that are currently used in a number of vaccines. Um, I'm married. My wife's a nurse. She actually was a COVID nurse for 3 years, taking care of tons of COVID patients in the ICU, and I have identical twin girls who are 16, and I'm always performing my own internal experiments using them to see are they actually really identical. But I always say to my friends, they're never good at the same time. So I wish they were, though. Be a lot easier on me. But wonderful girls, they're going into science. And my daughter is a researcher at the University of Houston School of Medicine, and she is working on RNA biology and trying to use RNA to technology to prevent congenital diseases. And she works in yeast, and I used to work in yeast at DuPont Merck, and she works on a similar pathway that I uncovered in mammalian cells about 25 years ago. So very proud of her. Okay, let me share my screen. Let me share my presentation. And can we see that? And here we go.
Yes, but do it in presentation mode.
Oh, I also want to make a little shout out to Brad Sturgeon. I noticed he's on the phone. He's a chemistry person, so you have a chemist on board today, and I'm collaborating with him on a few things through my consulting company. I also want to say that I'm a vice president of a biotech company called Cygnus Biotech. We're working on a host targeting therapeutic, and that targets in one form is a broad spectrum antiviral. In another form, it's a broad spectrum anti-cancer drug. So we may be seeking collaborations in the future with members of this group to help uncover the mechanism of action. So little preview. I also represent a ventilator company. You'll see a picture of their ventilator later in the presentation for procurement back to the government. And I routinely give talks on careers in science across the United States, high school, college level, sometimes even down at the grade school level. So I really want to promote science, promote our youth going into science, and taking over for us as we age and get older and get out of science. But I don't know if I'll ever get out of science completely. So, all right, here I go. First time through ever. I hope you enjoy it. But it brought back good memories when I started putting this together. So, all right. So here's the abstract that I was submitted. I'm not going to read the whole thing, but Neisseria meningitis is the leading cause of bacterial meningitis. It's in the mucosal cells in the upper respiratory tract, and then it's usually it colonizes probably 10% of the human population has some form of Neisseria in their upper respiratory tract, but they don't get disease, so they're carriers. And then this seminar will describe the bacteria, the clinical manifestations of disease, the pathophysiological and immunological aspects of disease that results in meningitis and provide information on how to protect and to treat the disease. Okay. So bacterial meningitis in general is a life-threatening and central nervous system infection that requires prompt diagnosis and treatment. The Neisseria meningitis can kill you within hours. Once it gets into the CSF, it's protected from immune response. If you don't have a great innate immune response to keep it in check, and you don't have any antibodies, you're not going to be able to fight it off. So, and then 95% of people with bacterial meningitis present with two of the four symptoms: fever, neutral rigidity, stiff neck, altered mental status, and headache. What happens is that the infection becomes bloodstream borne, and then it enters the meninges and infects the meninges that cover and protect the brain and the spinal cord. So that's where all its pathogenesis, a lot of its pathogenesis is in the central nervous system. So it's an aerobic gram-negative bacteria. Here's a picture of it. Gram-negative means that it has a loosely linked peptidoglycan in the outer membrane, and this causes it to stain pink under a gram stain. Gram-positive bacteria have stronger bonds, are highly integrated, and are not susceptible. Do not incorporate the dye, or incorporates the dye much better than the gram-negative bacteria. So there's 12 different types of polysaccharide capsules that determines the serogroups. We're only going to be talking about the ones that cause serious human disease: A, B, C, W, and Y. This polysaccharide capsule extends far off the outer membrane. It's a antiphagocytic protective mechanism by having this polysaccharide capsule, and it helps survive. The bacteria survive in white blood cells. It can be engulfed in, but these bacteria still can live, not at the level of in the bloodstream or in the CSF, but they could be re-released into the circulation or CSF from the white blood cells. It has an IgA protease that digests IgA. We all know IgA is the first form of protection on the mucosal surface of surfaces in your body. It carries plasmids that could carry antimicrobial resistance. It has fimbriae pili, which are very essential to the attachment of the bacteria to surfaces in the body. So here's the structure of the outer membrane. We have I call it lipooligosaccharide. They call it lipopolysaccharide here. The lipooligosaccharide is shorter than what you see in gram-negative pathogens like E. coli and Pseudomonas. It's more truncated, but it's very variable between strains within certain serogroups. So there are different linkages that make it of sugars that make it different from strain to strain. The outer here's the peptid glycan cell wall. It's loosely held together, more loosely than gram-positive. There's two proteins called pore A and pore B. They're the porin proteins, allowing for nutrients to enter the bacteria. And then there's the OPA proteins that facilitate bacterial adherence to epithelial cells and neutrophils. As I mentioned all the other characteristics before on the other slide. So we're going to move to the next slide. Oh, it also has a pilus. The pilus also allows attachment to cells. This has a lot of antigenic variation and could be hypervariable. So here is a schematic of how the bacteria enters the meninges and crosses over from the blood to the CSF. Oropharyngeal colonization, upper respiratory tract. Here's the diplococci shown here by, I believe this is an electron micrograph of the diplococci. And what it does is it attaches to these surfaces, these endothelial surfaces, mucosal surfaces, and that allows translocation or cytotic passage across the tissue. It enters the bloodstream from the mucosal barriers, and then it goes into the CNS, crosses the blood-brain barrier. Now, when I've looked in the literature, this is a dynamic process, and hasn't been fully characterized. This bacteria grows the best in humans, so it's really a strict human pathogen. So what makes the meningococcus able to infect is it has genomic instability, and then it allows for adaptation to the physiological environment. It uses different mechanisms to change its DNA. It mutates and actually changes its whole genetic profile during an infection. So let me talk a little bit about that. There's 4 ways in which it does that. It's through phase variation, where different proteins are synthesized, and there are different start and stop sites, and there are different genes that can be used for making the proteins. So different variants of the genes. There's also a process called recombination. So it can take up other external DNA, and also bring it into the cell. Oh, sorry, that's number 3, horizontal gene transfer. But so recombination is the process by which parts or all of the DNA molecules from 2 separate sources are exchanged or brought together into a single unit. The pillin and the lipooligosaccharide, multiple genes for them, they do the recombination within the genome, and you get production of a different sequence for pillin, and also a different type of lipooligosaccharide. So it uses different enzymes to piece together the oligosaccharides to form the lipooligosaccharides, which are shorter forms than lipopolysaccharides, but still have multiple oligosaccharide residues to form its structure. So then horizontal genes transfer from exogenous DNA, could be other bacteria that are co-infecting or co-colonizing. And then there's this process called mutators, which allow for more inefficient DNA repair, differences in the replication fidelity, and then these overexpression of DNA translation polymerase that may contribute to increased global mutation rates. So wherever it exists in the environment, and mostly in humans, in the population, will affect how it's transformed to a new entity, basically. So there's a lot of variability in these, and also Neisseria gonorrhea has the same type of process to mutate its DNA. So what's the response to meningococcal infections? Well, there's a inflammation process. There's these things called pattern recognition receptors, and also pathogen-associated molecular patterns. So in the host, the PRRs are toll-like receptors, nod-like receptors, C-lectins, scavenger receptors, pentraxins, and others. The PAMPs are usually the lipooligosaccharide, peptidoglycan, and the outer membrane proteins and metabolites. So this allows for an immune response that generates cytokines, similar to what we've seen with COVID. NF-kappa B seems to be one of the major regulators in the expression of these inflammatory genes, and things like TNF, IL-6, IL-1 alpha, IL-8, CXL10, CCL5 are produced. So in some forms, these immunomodulators or cytokines can be good for the body, and some ways they can be detrimental. So as we've seen with COVID, the expression of these are detrimental, especially in the physiological conditions in the lung. So the TLR4 is the major determinant that drives the pathophysiology of invasive meningococcal disease, and it's also crucial in prevention of it. So, so it's not quite really understood on when and where TLR4 is able to do its positive things or its negative things. So it just depends on the infection and the strain and the serogroup of bacteria that's infecting. So we also generate humoral antibody, and that's the neutralizing immune response. I don't think that a productive um, meningococcal infection can be totally cured using an innate immunity. So we use neutralizing IgGs developed to the capsular polysaccharide, and that allows for phagocytosis and also elimination of the bacteria from the body. Infants and children recognize capsular polysaccharide in a T-independent manner and only make a weak non-neutralizing IgM immune response with no memory. So these conjugate vaccines that have been developed allow for the child to recognize the capsular polysaccharide in a T-dependent manner, which allows for IgG production and protection for a number of years from infection. So these vaccines are conjugate vaccines. There were vaccines out there for adults that were only capsular polysaccharide, but now everything has gone to the conjugate vaccines. Other vaccines that are conjugate vaccines that are licensed are vaccines like Prevnar for streptococcal pneumoniae. And now Prevnar is up to 20 different serotypes that it protects against. I worked on the Prevnar vaccine back in the 1990s, and the first licensed vaccine only contained 7 serotypes. So the currently licensed vaccine contains 20 serotypes. So this is the whole infective process in a nutshell. I'm not going to go into too much detail here, but I want to make you understand that there's things like complement activation that allows for killing of the bacteria. There is the process of the bacteria entering the cell through phagocytosis. There's also the stimulation of the inflammatory immune response off of NF kappa B transcriptional regulation. But if you look in this picture here, the dotted lines, the dashed lines are not fully understood. So the take home message here is we do not know everything about this bacteria, how it infects, and how it causes its pathogenesis in humans. So, how do we protect against meningococcal infection? There are 3 vaccines licensed in the United States, one by Sanofi and 2 by Pfizer. There's a vaccine that contains all the serogroups that cause human disease. B was the hardest vaccine to develop. To the B, for so many years, we did not have a good vaccine to serogroup B. And actually, in my research, I worked on serogroup B strain at Baxter. So there are 3 routes you can get vaccinated for meninge B. If you just get 2 doses 6 months apart, you're protected. But what Pfizer has developed is a vaccine where you immunize with all 5 serotypes, one immunization. um, one immunization and then boost with the group B 6 months afterwards. So these are two ways that you can get vaccinated. They can be administered between the ages of 10 and 25. Again, um, and then the one vaccine, the Sanofi, is given twice at about 11 or 12 years and then again at age 16. So we're trying to protect mostly people that are in close quarters, are adolescents, people, and I'll talk a little talk about people who are more at risk as for meningococcal meningitis. Again, the leading cause of bacterial meningitis and sepsis in the U.S. Those vaccines are these vaccines are licensed. Um, it also can cause pneumonia and focal disease such as septic arthritis. Again, this is just a repeat of what I just discussed. So the symptoms are sudden high fever, stiff neck, bad headache, nausea or vomiting, confusion or trouble concentrating, seizures, sleeplessness, sensitivity to light, no desire to eat or drink, and then a skin rash. So when you look at this skin rash, this is called the Blanche test, which I never even knew about. So generally, with some rashes, if you put a glass against them, the skin will turn white and you won't be able to see the rash anymore. However, with the meningococcal rash, when you put the glass up against it and press against it, you can still see the rash behind the glass. So this is one of the indications that it's a meningococcal infection rash. However, we need faster diagnostics and we need more specific diagnostics because a lot of these symptoms can be confused with, say, influenza or COVID or a number of infectious diseases that cause similar symptomology. So we're going to talk later about the diagnostic tests. Here we go, the diagnostics test. And actually, I worked in the stat bacteriology lab at a county hospital in Buffalo. I worked there part time while I was going to graduate school, a couple nights a week, and I had to actually perform these analysis, take cerebral spinal fluid and do a gram stain and do a bacterial latex agglutination assay, and so the diagnosis is doing the major rapid diagnostic is actually doing a assessing the spinal fluid by lumbar puncture and taking that and sending it down to the microbiology lab, doing the gram stain, and you can actually see in this leukocyte, the diplococci right here. In the cerebral spinal fluid, it's very difficult to actually see the bacteria because it's not heavily populated in the CSF, and you're only taking one little section when you do the one little amount when you do the gram stain, and it's very difficult to actually visualize diplococci in the gram stain. It's much easier to have it concentrated in a white cell to visualize it under a microscope. But then you do this antigen test, and this antigen test is based on antibodies that recognize the capsular polysaccharide. And when you see agglutination here, you know that CSF contains this strain of this serotype of meningococci. So that's the positive result. This is the positive control, and these are negative. And I've actually done these assays, and actually, several times we're able to identify what was causing the infection. So doctors usually empirically put patients on antibiotics even before they get these test results back if they think it's meningitis. So they also do imaging, and that shows also uh, the disease, but that takes a little longer. and may not be highly indicative of a Neisseria infection like we see with the agglutination assay. And then they're cultured, and they grow on blood agar. And then there's other tests to identify them based on their metabolic enzymes. So these are the three ways that we diagnose bacterial meningitis. This is the treatments. Mostly, these are all IV treatments, and pretty much similar to what you would treat an adult, newborn, and child over two. There are a number more different types of treatments you can use for adults. So, but basically, these are the treatments. So, what's the supportive care? The managing and clearing the airway, the mucus and everything in the upper respiratory tract, maintaining oxygenation, using supplemental oxygen, giving sufficient intravenous fluids, and providing fever control using Tylenol or ibuprofen. And if it gets really bad, you have to be put on a ventilator. And this is just a picture of a ventilator. So you don't want it to go this far. So, all right. Let me talk a little bit about what we did at Baxter and what our research focus was. So we were developing a meningococcal group C vaccine. We also we're developing platforms for delivering other conjugate vaccines. So we were studying the porin proteins of Neisseria meningitidis. And in particular, we were studying Group B and Group C Neisseria meningitidis strains. So a colleague of mine in the laboratory was studying the structure, the physiological structure, the properties of the class 3 POR B protein, and she did studies on that, and we ended up publishing a publication. So we were able to actually show and compare and write a paper on the differences between class 2 and class 3 proteins, and show that they were really not that much different. So POR B has a minor role in antibody influx, where the loss of this protein decreases susceptibility to the antibiotics. And then also, very interesting, we were working in the laboratory and we showed that POR B was able to catalyze the polymerization of actin, which is very interesting because the bacteria, when it enters the cells, the phagocyte kind of walls itself off from the destruction of destructive enzymes of the phagocyte, and perhaps the polymerization of actin allows that to more happen, so the bacteria can survive more in a vacuole within the phagocytic cell. So this was published in 1998. And I'm not a protein chemist by no means. So Cisa Menetti was doing all this work in the laboratory, and I just provided her purified protein. We expressed this protein very easily, recombinantly in E. coli, and it was centralized or expressed in inclusion bodies. And we were able to isolate very pure porin from these inclusion bodies, and then um, by passing it over columns and refolding it to form a very well defined structural protein that formed a pore when we put it together in solution. So, what we wanted also to do was to be able to purify the porin protein from the wild type meningococci and compare the two. So it was very difficult to isolate the porin protein because a protein that was very similar to the AMP protein in E. coli was contaminating our preparations. So I was asked, I was asked to design and knock out the AMP-like protein in the Neisseria meningitidis strain, Group B strain that I was working with. I think the name was BC4 was the strain. So my work, I developed a one-step method for genetic transformation of non-pileated Neisseria meningitidis. So the issue here is that these bacteria um, are these bacteria are reduced in the uptake of foreign DNA. Number one. Number two, um, they're not very easily transformable. If you do electroporation, there are no surviving bacteria. So electroporation, as you know, is an electrochemical method of introducing DNA into bacteria, and it's done, under high voltage. You actually the cuvettes you do it in, you have to actually put them on ice first because there's a heating up of the solution in the cuvettes that could actually kill the bacteria. The meningococci have to be grown in special media and cannot be and in glassware or plastic flasks that do not have any detergent. They're very susceptible to detergent. So what I did was I was able to actually look in the literature and find a transformation protocol for E. coli. It was a gentle heat transformation, and I was able to adapt that protocol using our media and for growing the meningococci, and we're able to actually knock out the AMP-like gene in uh, the strain that we're working in, and then that strain was then used by CISA to isolate the native porin B protein, and she compared it to the recombinantly expressed protein and was able to show that they were structurally identical. So we also were working on a Neisseria meningitidis Group C vaccine that was covalently coupled to tetanus toxide. We developed it at North American Vaccine and Baxter, and it was licensed in the United Kingdom in 1999. The Group C polysaccharide is covalently linked to tetanus toxoid. It's supplied in a pre-filled syringe without a needle, containing one dose in half a ml. Each dose of the Neisseria vaccine contains meningococcal group C polysaccharide conjugated to 10 to 20 micrograms of tetanus toxoid protein with aluminum hydroxide, sodium chloride, and water for injection. In 1999, the UK introduced these vaccines from several companies in all infants in 3 doses at 2 months, 3 months, and 4 months of age, and in one dose um, in children 1 to 18 years of age to reduce meningitis and incidence and death. So there sort of was like a mini epidemic of Group C meningitis in the United Kingdom. So there was hundreds of cases, and there was a number of deaths associated with this serogroup of meningitis. So I just want to show you what we were able to accomplish through this immunization process in the United Kingdom. So the next slide will show that. So here's the menin C cases from 1998 to 2022. And as you can see, when we were starting, there was almost 1,000 cases a year. And by actively immunizing children in the United Kingdom, we have brought that incidence rate virtually down to zero. There were deaths here. There was probably up to 50 to 100 deaths from meningococcal disease and that went down basically to zero within a few years. It went to zero, totally no deaths from meningitis C. So this is what you call a program. This is really what you want to do. You want to bring the cases down to zero. Now, we have a vaccination program for COVID. Our vaccination program for COVID has not brought the cases down to zero. So I always say, if you can't do that, you need to get a better vaccine because COVID is more easily spread than meningococcal C, right? So you're going to keep that level of infectivity uh, around almost forever if your vaccine is not uh, durable and shows protection for lifetime. So it's very important when developing vaccines that we get a handle on that and improve our vaccines to be just as effective as what you see here. Okay. So, what are the high risk groups? These list all the high risk groups: medical conditions, complement deficiency. Um, if you don't have the complement to um, bind onto an antibody bound to the bacteria, then you're not going to destroy the bacteria. Um, um, HIVs, immunodeficiency. Um, if you're on complement inhibitors, that's another way that you could be more susceptible to meningitis. Different settings, traveling to or residing in countries where these strains are common and cause a lot of disease. A microbiologist. I actually know two microbiologists that were exposed to meningitis in the laboratory, and one of them died and one of them survived. So that's 50-50 right there. So you have to be very careful when working with these strains. Um, a military recruit, basically in living in barracks, uh, allows more efficient spread of those who are colonized with the bacteria to other people. Living in a residency hall and not up to date with the vaccine, and then also being part of a community experiencing a serogroup ACW or Y outbreak. So this is an example of a hot meningitis belt in Africa, and this just shows relative number of cases. And as you can see, in the center here is where they have more higher cases than the other areas. And I believe this is meningococcal A is the most prevalent serotype. In the United States, it's actually meningococcal group B. United Kingdom, it's meningococcal group C. So depending on where you live, you may be facing a different serogroup. All right. So the key take-home messages from this part of the talk is that meningococcal disease is serious, uh, and it can be deadly within hours. Early diagnosis and initiation of antibiotic treatment is very important. People spread the bacteria through close or lengthy contact with other people, as demonstrated by the military recruits and the college students in dormitories or fraternity houses. Anyone can get meningococcal disease, but certain people are at increased risk. And I went over the types of environments and people that could be more to have more risk to get infected. And then vaccines offer the best protection against meningococcal disease. So I'm older. I've never been immunized for meningitis. The vaccines weren't out until after I got out of college. I also have not been immunized with Haemophilus influenza type B. I also have not been immunized with streptococcal pneumonia because those vaccines were developed after I was an infant. So a lot of us have not been vaccinated with these vaccines that are out there. I think that my next shot at a vaccine would be getting the Prevnar vaccine at age 65, and these other vaccines I probably are likely immune to Hib and to meningococcal infections. So, but our younger folks who are in those environments are not, and that is important that they get vaccinated. I believe the meningococcal vaccine is required for college entry in the United States. Maybe not all colleges, but a good number of colleges, and I believe a lot of this is mandated by each individual state on what their requirements are for meningococcal vaccination. So I Raj brought this up to me before the meeting, and I was not aware of this publication. But there is a process in which the bacteria is able to cleave complement, facilitating degradation. As we know, complement C3b uh, gets deposited on the bacteria, and then the macrophage or phagocyte has C3b receptors, and that allows for phagocytosis and destruction of of the uh, the bacteria. So this is a publication about, I would say, 10 years ago, and that this bacteria makes a enzyme that cleaves complement to inactivate it. So does anybody want to discuss this, or Raj, you want to talk about this publication?
You are muted. You are muted.
Okay. Thanks, Dr. Bogdan,
You're welcome.
for a nice presentation. We want to understand this system, so that's why when I saw your resume for KSAS Advisory Board, so for all the people, Dr. Bogdan is also in our Research Advisory Board of KSAS. Uh, so I saw his resume and I found that he has some publication uh, on Neisseria meningitidis. So then I thought that we were doing some project related to this, in which Abhinandan is trying to do. He is also in this meeting also. So he is trying to to cleave the complementary system, and because normally the complementary system C3 is when it cleave into C3A and C3B, then it tag the uh, injured neuron, and then macrophages and other astrocytes and glial cell attack that particular injured neuron. So, but this protein from the Neisseria meningitidis NAIP is cleaving the same C3 protein, but on just 4 amino acid before the actual cleavage site in the normal physiology, and that makes it inactive. So it is not able to attack the neurons, and also not bring the inhibitory environment for regeneration of the neuron. So that's the project we were trying to do it KSAS. So that's why I requested Dr. Bogdan, and he is very kind to accept my invitation to present on this. And he really presented very well on these topics to understand us better about this bacteria. Uh, he probably does not have too much time to discuss about the other protein which this bacteria produces, which can be used as a therapeutic molecules. So although he used one protein in this pro-B, which is shown to be catalyze the polymerization of actin, and that could be a very good target for actin polymerization in the injured neuron. So uh, this is just the background how we got to this discussion.
Yeah, I think for this project here, we need to get a little bit of handle on what the differences between the convertase and human convertase and this enzyme here, if there's any similarities. but developing small molecules and stuff to this protease may be easily done if it's significantly different from what's been studied before. So my thing is you have to develop an assay and be able to screen libraries of compounds for inhibition of the protease. And I think that would be the path forward here. I am not sure if you target it with an antibody because it's probably an internal and secreted protein that may not work. It's not going to work to kill the bacteria, but since it's probably secreted, um, you'd have to inhibit it in its isolated form. Um, uh,
Yeah.
maybe
So
the antibody, and we have problems with antibodies crossing the blood-brain barrier. So if that's where its activity is more significant, then you would need a small molecule. Um,
yeah, our target is not the kill the bacteria. Our target is to uh, prevent
Neutralize the protease, right?
prevent the degeneration of the neurons.
Well, it's if you prevent, is it? This is this is this is still targeting. So when it cleaves, is it still active or no? Is it
No, it's not active. When it this NAIP when cleaves C3 protein, it's not active to tag the injured neuron or degenerating neuron.
okay?
So that prevents the attack of all the glial cells and astrocytes to the injured neuron. Then we can target that particular in neuron with some regenerating molecules.
Regenerating molecules. Okay.
So we have like BC3E, which is regenerating the neuron. So that molecule can be more effective in that case when we stop the C3A and C3B, active C3A and active C3B formation in the astrocytes.
Okay. So you would want these protease to actually do that within the neuron?
Yes,
You
within
would
within
want it to do that. You would not want to inhibit it from doing that, right?
yes.
Okay, I get it now. So how big is this protein and can you recombinantly make it and use it in conjunction with your
Yeah,
regeneration?
yeah, we are synthesizing right now.
Okay. So, and then you'd have to develop an activity assay with C3b, right?
Yep. Yep.
To determine its full function, right?
Yes. Yes.
So you'd be doing similar things to what my colleague did. You'd be doing the refolding and determining the topology and the structure of the enzyme and everything like that. So they're all techniques that can be easily done, in a laboratory setting. Um, so what happens? Have they generated any knockouts of this enzyme in meningitis strains? Have they knocked this gene out?
No, I don't see. There is not much publication on this. This is the recent
Yeah.
publication. Abhinandan, you want to
Well,
comment
if
on
you ever
this?
want to knock the gene out, I know how you knock it out because I have the transformation procedure. So,
Yeah, yeah. So I think
No,
only one or
act,
two
act,
paper on this protein. So
yeah,
actually, actually, in this paper, they characterize this protein and they validate their function. This paper published in PNAS, I read this paper, and they only validate the function and they found that if they mutated this protein in the bacteria,
yeah.
that bacteria, they they decrease the severity of the disease.
You reduce the
Mr.
severity of the disease.
Yes, yes.
That's makes sense to me.
Because this protein help bacteria to acclimatize in the brain environment because it
Yes,
destroy the
yes.
complement system.
What about the blood? What about in the blood? Does the bacteria
No, they did. Blood.
is
I
there
think they
blood?
did
Is
it
the
with CSF.
bacteria able to replicate as well, to divide as well, or grow as well in the blood with this enzyme knocked out, or is it the same?
No, I think they did in the CSF only, and there they
Okay.
Yes, I
were,
think so.
yeah, the CSF they were growing. These bacteria were
Okay.
growing. Yeah. So as you said that
Well, if it also does it
bacteria
in the
usually,
blood too, then you have less crossing over to the CNS too. So it'd be nice to see in blood, does it do the same thing? So, um, because that's where the bacteria is coming from.
yeah,
The bacteria is higher. I'm sure. I'm positive that the level of bacteria is higher in the blood than the CNS. So,
yeah.
I mean, I would think so, but it's been a while since I worked on this whole thing. So let me move, and I don't have that other reference here. Um, I can pull that reference out for everybody, or the PDF, and send it around. So here are just the references. I mean, some of this is information you can find at CDC's website. Um, I looked at a paper on the immune recognition, and I also looked at the genome instability and DNA repair. Um, this is a pretty good publication in neuroscience. This is an excellent review. Um, and then these are just basically sites that anybody can go to and pull out information. And then here are the publications that came out of our laboratory at Baxter Healthcare. So, so North American Vaccine was a startup biotech, and the story there is that they bought they bought they bought North American Vaccine for their meningococcal C vaccine. We also had an acellular pertussis vaccine, and we're working on other vaccines for Group B strep, and in particular, we were going to use the porin protein as a potential carrier molecule for a Group B strep vaccine. So that's how we got all involved with POR B. We also did experimentations using it as a carrier for the meningococcal C. Um, Everything within the same bacteria, the POR B and the C polysaccharide, isolated, recombinantly made or isolated from wild type bacteria. So that's how that all got started. Then Baxter was able to get the contract with the United Kingdom for like $65 million. They, you know, were making GMP product and shipping it over in filled syringes that they only needed to put a needle on and inject. Uh, and then they had a viral vaccine division in Austria, I believe, working on tick-borne encephalitis virus vaccines and also influenza vaccines. So after they did this deal and everything like that, a few years later in 2002, they decided to lay off or get rid of their whole bacterial research division. And then in my career, I needed a job, but they were paying me for like 9 months. And within a month or two, I wanted to join the NIAID because they received $3 billion for starting their biodefense program, and I thought it was a great opportunity to get in there uh, and work on improving the infrastructure. And I worked on containment lab applications, all the regional and national biocontainment labs. I helped set up the reviews, and then I worked on product development. And I think the major accomplishment there was developing monoclonals, a licensed monoclonal to anthrax, and then a six monoclonal antibody formulation to botulinum neurotoxin, which I worked on at NIAID, and then I was called back to work on at the Department of Defense at Fort Detrick. So that was the last position I held with a company or a organization. And this July, I left and went back to a business that I had started about 10 years ago. And now I'm doing um, consulting. And if you need any consulting work, I am available. I'm an expert product development. I'm an expert in federal funding opportunities and partnering, and generally an expert in a number of different disciplines of science, from bacteria to viruses to toxins and diagnostics and vaccines and therapeutics and probably a few other things. So if you ever need any assistance or um, want a little bit of advice, I always say that my first hour is free, like a consultation, like a lawyer, right? So, but thank you very much for giving me the opportunity. Thanks, Raj, Balram. It's a pleasure speaking to an international audience. I usually just talk to the people in the US, but I said, well, now I have a little international outreach is always good. So, and thanks for this organizing this whole different way of addressing science. Um, I think and I believe in the spiritual aspects of things. So I think that sometimes a little prayer doesn't hurt, and I pray that everybody's experiments work. So thank you very much.
Thank you.
Okay,
Thank you. Thank you,
I see we have a
Bogdan.
nice audience. I see we have a nice audience. I didn't see all the people. Are there any specific questions? Did we get anything in the chat? Let me look at the chat.
I have a question.
Okay.
Yeah, so I was discussing over the phone also, and to you, that is there any other protein? So one of the protein I found in your presentation is ProB. So do you know that the ProB structure and the catalysis mechanism is known, or they are doing through the GTP GDP mechanism of the actin polymerization, or they are doing something else?
No, not to my knowledge, no one's really working on this, and this is something we uncovered through another investigator that was working in another laboratory, a more academic laboratory. We do know you can actually knock out pore A and B, and the bacteria still grows. So it's not essential for the growth of the bacteria, but it might be more essential for the pathogenesis of the bacteria than growth and survival. So that's where it stands. She did the structural analysis. These proteins are really stable. They form similar structures, whether it's a pore A or a pore B, very similar in structure and function. So they could be potentially a target, and they could be a way in which the bacteria walls itself off from the immune killing of the phagocyte. So that's what we were thinking about when we were doing this
Yeah.
program.
So I was thinking not to kill the bacteria. I was thinking to use this molecule from this bacteria as a therapeutic molecule for initiating the actin polymerization.
For catalyzing it.
Yeah. Yeah.
Well, you have to figure out what disease you think it's going to work in and stuff like that. I mean, I don't know. Yeah. So what disease would it work in? Is it so muscle actin polymerization is actin myosin, and it could be involved in some
Yeah,
disease.
in all kind of degenerative neuron disease, this
Yep.
depolymerization of actin is one of the concern, and because of that, especially in the CNS neuron, because
Yeah.
CNS neuron normally they don't after injury they don't produce the uh, actin polymerization or actin reorganization as efficiently as in the healthy neuron, and that's because of that the injured neuron form blob. Instead of a synapse, they form blob, and blob
Yeah.
is crisscross of all the actin filaments. So because of that, so if we can initiate the catalysis of actin polymerization, then they will start reorganizing themselves, and then neuron can grow further, or action can grow further.
So I do
Yeah.
remember we did localize it to a peptide region that allowed for the polymerization. So it's not the whole protein. We were actually able to go down to like a 20 amino acid sequence or something like that. So more in the small molecules. So, but I am not sure where that information is. It's been so long ago. But I believe I actually have buried away. I probably have it in a box. The sequence of the porin protein, how it loops through the membrane, and also the region where the peptide region where that catalyzes the polymerization. So we did some studies using deletion mutants and expressing it in E. coli and then testing it against actin.
Okay.
So yeah, there's a region probably in the porin that can be used to catalyze actin polymerization.
Yeah. Okay. That would be helpful.
You don't need the whole protein.
Yeah. Yeah. Oh,
I think the protein's like 40,000 or something like that. I don't know. It's pretty big. So, but if it forms the pore, So, any other questions?
John, I have a question, or maybe you just said about something about philosophical, but before we go there, I was just wondering if you have paid attention to using the mechanism by which it gets across the blood-brain barrier, which is, it seems like it's using cytokines in addition to infecting the endothelial cells. How much is known that we can use sometimes to deliver some other drugs, something else other than this is
Yeah,
delivery?
that's a tough question because these bacteria are, listen, in the bacterial field, not enough stuff is funded. Not enough stuff is known. I think the cellular biology field is out advancing the prokaryotic field because there's just not the number of investigators out there. There's not enough funding to do these critical studies. You get a vaccine out there for meningitis, right? Well, and you have antibiotics that can treat it, then what's the initiative of the federal government to fund research and when you have a cure for it or you have a drug for it? So it's it loses its flavor, right? And then you loses its funding, and then it loses its traction, and then it loses eventually the investigators retire, and then the programs just fall away. So it's very difficult. I believe that these things should be researched. I believe it's important. The Neisseria is a unique bacteria, does things that other bacteria do not do, and it has mechanisms of protecting itself, and it's this antigenic variation that allows for it to survive in different environments, and it changes. It's got antigenic variation, although the capsule does not change. So that is an appropriate target for the vaccine. It's susceptible to antibiotics. It's highly susceptible to antibiotics. So I mean, so it's how do you get the funding to explore this question? That's my question, and it's very difficult to get that type of funding.
Yeah, I think that's a good question, and I think, I mean, good point to make. And as you said earlier, that this group is a very, very tiny effort in
Yeah.
trying to change that. So we need to create our own funding. We, I mean, I know you have connections to government and other agencies, and
But
maybe
it's but guess what? You know, going back to working on something you worked on before, like I went back to monoclonals for botulinum neurotoxin, can be productive, right? So, I wish
so.
I had
I
more
wanted
time.
to.
I wish
I
I had
wanted.
more time at Baxter to spend on answering those questions because I could have had a long career at Baxter just doing this type of stuff. But once they solve something and they sell it, guess what? They kind of push you out the door, right?
Yeah, I know. I know that I heard that you just missed the meeting. I was there for the last couple of days in Atlanta, and where I met at least half a dozen people who, like you are saying, who have a lot of cumulative experience, and they are in a situation where they do not make those many mistakes, and then can move things forward. But then, like you said, when you are out the door, then that whole thing, it just goes down the drain. So,
So the
so
better you do, the worse off you are. The less better you do, you keep a job. I don't
yeah,
know, right?
yeah.
Well, there
So
are other opportunities. So I get to work with you, and I get to work with
yeah, yeah,
the
and
other company I'm host targeting. I get to work with the ventilator company, and then perhaps a vaccine company coming up. I, it just depends, and then different doors open because mRNA vaccines. Well, we need to improve them. It's okay. They worked initially, plugged the dike and kept COVID levels down. But guess what? They're not the total answer in the future. I mean, just like the single polysaccharide vaccine with no conjugate to meningitis C was not the best, was the answer at the time. But now we got into this conjugate technology and everything's moving in that direction because you can give it to infants and adults and it still works, right? So you're on the same platform. You don't have multiple, just polysaccharide alone or polysaccharide conjugated to a carrier molecule. They're all moving to a platform that's simpler and easily reproducible and manufacturable and marketable. Right? It's like going to Walmart. You can find one thing on the shelf that solves all your problems. You're going to buy it, right? So, and then going in depth and trying to figure all this stuff out. I mean, hey, guess what? There's a lot of neurological diseases that are not really fully known and researched. And I thought like ALS or Parkinson's or that type of stuff. Maybe some of these things could be used to help treat. So
yeah, but what I was saying is something you said. Well, first of all, something you said, that your experience, and I was just saying there are such um, thoughts that I heard this time from several other people, including Eric Johnson and others, that they are like shut out. Once they retire, they're just gone. And for this group here, and what we want to do in the future with this kind of activity is to find a way to inspire and train the
yeah,
new group of people, young people, who learn a little bit different than what you and I may have faced because of
yeah,
our experience, if we use our experience well. And that experience is that, once you retire, you leave your position, you may have all the experience, but then, it's no use to it. So what we want to do is, I offered several of those people, including international, very experienced, very knowledgeable people who were thinking that their life is over as far as professional thing is concerned. So we want to build, like KSAS has a lab, and there are young scientists and philosophers and others. And I'll come to that philosophy because you just said we all have to pray. then it doesn't hurt to pray because at
Hey,
least it shows the humility.
we need to rejuvenate ourselves, and when we rejuvenate ourselves, we rejuvenate the young. We get them moving forward.
You-
I usually talk my start of every talk when I go to careers in science seminars that I give is the reason why I got into science is not because of the money. It wasn't because of the fame. It wasn't because of, you know, anything like that. It was because I wanted to save lives. And I said to myself, John, what is the best way that you can contribute to society and save lives as a scientist? And that's developing vaccines, therapeutics, diagnostics, stuff like that. It's all about saving lives in the end. And I think that's what we need to teach our youth. We are here to save lives. We are so important. A doctor would not be able to prescribe a medication or a vaccine unless we developed it, unless we saw the human need for it, and we're altruistic in our efforts. We are altruistic because we think and live and breathe this every day of our lives, and it's all about saving lives. That's what it's all about. So,
Yeah, well, what I was saying is that in addition to the fact that you can get young people trained and excited, but also because of them, we also get very rejuvenated because we have now think about something new.
yeah,
And um, and then which the new thing is here is the what I want to say that here is a organism, a bacteria that has not been that well studied, despite the fact that it has been a big problem for a very long time, and many of us, including myself, my father passed away because of the tuberculosis, actually. And the tuberculosis also causes meningitis, believe it or
yeah.
not. So, but we have not learned that much because there is all kind of other pressures. So when I think of it, I did make a presentation where I said we think we are very smart, and I think we are not. This bacteria is more smart. It has figured out. It has more intelligence, so-called. Talking about artificial intelligence. Here is a natural intelligence that it beats us every time we try to do something, and so from that point of view, just from the point of intelligence or whatever its own mechanism is, we don't even understand. We do not understand even the question that I asked about how the mechanism can be used. We don't even understand that. So
Yeah.
just imagine that it does day in, day out, the different kind of intelligence that shows us. So we need to be cognizant of that as well.
Well, maybe the phagocyte helps bring it across and then it just comes out of the phagocyte. I don't know. I mean, that might be a way because you have this translocation and those cells know how to do it, right?
I mean, we learned so much for botulinum toxin, as you
Yeah,
you you can see, that we have created a few things that are very helpful,
yeah,
including now we have oral vaccine created because we learned how the botulinum toxin is a food poison. How does it evade our digestive system? We learned that, and then now we have 3, 4 vaccines which are oral, otherwise not available. That includes even tetanus and COVID also. So the point I'm trying to say is, in a person like you who is who talked about your family, young family members, and how rejuvenating they could be for you. I think this young group of upcoming budding scholars, future scientists. I think to me, I feel the same way for them. So if we can find the experienced scholars like yourself with so much interest in young people,
yeah.
starting with the family and then the students and others. I think there is a very, very good hope.
Well, the thing is that guess what? When we were first starting out, us older folks, but I consider myself young at heart. We didn't have all the instrumentation and technology that's out there today. We didn't have the computer power. We didn't have stuff like that. We were pouring gels that reached the ceiling and sequencing with P32 and P33, and we were actually designing our own protocols and experiments and making our own reagents from scratch, right? It's a it's a different type of science now because it's all out there. We just need to effectively utilize it and for these younger investigators and get them in the right spot to do the right work, at the right time. And they have the reagents, they have the equipment. We have a lot of knowledge already. It's just getting them to find a place to build on that knowledge. And before it was like, how do I have to make my own buffers up? And now it's like the buffers come, you can order them. I have to do my own sequencing. No, I'll just ship it out overnight. They'll do it. I'll get the sequence tomorrow. It's just another different game. We're in a different game now. It's a different game, you know.
True.
So
Somebody has to translate that different game.
they're trying to change baseball and football too to be a different game, all the different new rules and all that stuff like that. So guess what? Those games are going to eventually be different games, right? So,
You
but,
should you
but
should
It's
you
not
should also plan to visit India, John.
I
You should
will.
you should plan to visit India.
I'm working with a company, and their senior consultant was an ambassador to India,
okay.
but
Why
not
why
from the
don't you come
U.S.,
to
from another country. He's an ambassador to India, or was an ambassador. So I'm hoping to go visit him.
Sure, that will be great.
Yep.
why
So,
don't you
but
come?
find me a meeting to go to. I got to go for a meeting though, and I don't speak Indian, so I have to. It's all in English, right?
Well, I think there is enough people who know
Okay.
English in India, although your buffer system is translated a little bit different in India. They
Oh,
have these days they have started doing buffet for serving, and in the villages they call it buffer system. So you
okay.
don't get confused.
So yeah, but I would love to pick up a little bit of the language too and the customs and stuff like that.
Sure.
So I love Indian food. So
Absolutely. Well, thank you. Thank you
well,
very
thank
Why
you
much.
why
so
why
much. I
don't you
appreciate it.
join us?
Let me know if there's another topic. Maybe we'll do a little pre-meeting next time so I can make sure I incorporate everything into the presentation. But I think I did a good job at the last minute, and I think I was going to touch on a lot of the things that you guys wanted me to touch
I
on.
think
But
we will. I just thought about it because you mentioned about how you like to educate students. So we have a high school, and those students came and then worked in this institute there, and in the there for I think 2 or 3 weeks, and from what I understand from our science colleagues, that they did an excellent job. So we will invite maybe next time those students, and you can use some
I
of
give
your
my talk
experience.
on careers in science.
Yeah, career in science and what kind of things are available to them.
That's what I can do. That I just gave one to the Westminster. Really, my Carroll County, the county I live in Westminster, is a tech center.
Mm.
Now I got to tell you this story because this is a crazy story. I contacted them because I wanted to give a talk. They have a biotechnology 2-year program, high school. They got full laboratories, equipment. They clone DNA. They grow stuff up on agar plates. They do electrophoresis, Western blot. Those mostly column chromatography. So I gave a talk on careers in science, right? So happened that the person in charge I worked with at Wyeth Letterly 20 something years ago. She worked in another department. I didn't really know her, but we worked together. And then her colleague who teaches the other class, they have two parallel classes. I gave him a tour of Baxter Biosciences laboratories when he was a student at Towson about 20 years ago. So it is a small world. You get to meet people that you met before. It's a small world, and re-engaging with former colleagues is also the best therapy too, right? Keeping
So
those
true.
connections and re-engaging, that rejuvenates you, invigorates you as you become an older scientist or product developer or whatever. So you never know who you're going to meet.
I call older scientists as the scientists who have longer experience of being young.
That's true. So, well, thank you very much. I appreciate it. As always, let me know if you want to, if I can do another lecture. I love putting these together. You know, you got my, you know what I'm good at and stuff like that. So, it's been
So
fun.
if you want to come to India, then come in February. We have a symposium there, 3 day symposium. You you can meet several people there, and as well as you can see our lab and interact with the student as well.
Send me the information.
Yeah, sure, I will.
All right. I appreciate it.
Okay.
Thank you.
Have a great day, everyone.
Thank you.
Thank you.
Bye-bye.
Take care.
They will have great night. We will have a great day.
I actually, if anybody wants the Raj has copy. If I know you got that on YouTube, but Raj has a PDF copy of the presentation that he can share with you if you want.
Great. Great.
All right. Take care.
Bye-bye.
Bye-bye.